Are there any questions about what I was talking about earlier? So again, just remember that the really important biomarkers that we're interested in today are lipids and the composition of lipids and alkanes. In the next few slides, I will discuss these in terms of their preservation over the last really 400 million years, which is really remarkable when you think of organic material. If I do go a little fast, I think we're a little behind schedule, so if I pick up the speed a little bit, let me know and I'll try and slow back down. But in terms of taphonomy for biomarkers, there is evidence for exceptionally well-preserved biological material dating back to the Pennsylvanian and before that have been found in typically cold environments. So when these markers get preserved, they usually preserve better in cold or really waterlogged environments. When you have acid soils or really high temperatures, it's not always as conducive to preserving these molecules as some other locations around the planet. And I'll show you in a little bit, in really northern Canada, there's been some exceptional fossil finds that actually have organic residues still preserved after millions of years. So here's one example from a site in Idaho in the United States where these might look like typical fossils that you see or that you find, but this isn't a mineralization of the original plant. This is actual organic material preserved in these leaves. So the leaf structure is still intact. When they first discovered this site, the site's name is Clarkia. When this was first discovered in Idaho 30 or 40 years ago, they were clearing the land. It's a farm. And they were clearing the land with a bulldozer, and they kicked up a bunch of these organic residues, and they actually floated through the air like you would see with modern leaves. And that's really incredible when we're talking about organic. I mean, these are 15 to 20 million-year-old samples, and yet they were so well preserved that they almost look like, I mean, minus the coloration, almost look like modern leaves. And this is just one example of organic preservation. And from these materials, you can actually scrape this off, take a knife or whatever, and scrape this off of the rock and collect the residue. Or you can use microscopy, scanning electron microscopy, and really identify the characteristics that determine which leaves these were. So here's an example. Again, this is a metasequoia. A metasequoia is a dawn redwood. So if you're familiar with the sequoia trees in eastern North America, they grow up, 200, 300 meters high. This is a relative of that. It's considered a living fossil because before, 100, 150 years ago, we only knew metasequoia from the fossil record. And then a small population was found in central China, and it was exported around the world. And now it grows in multiple spots throughout the world. But you can see here from this 15 to 20 million-year-old fossil, and this is a modern one growing from the state of Rhode Island. They almost look, really identical, which is remarkable. So today, a lot of research has been going on, focusing on the organic preservation plus the micromorphology of these plants to get an idea of Miocene and Eocene climate changes over the last, from 50 to 10 million years ago or so. That's large scale. Those are really extreme examples, right? It's very difficult to find organic residues, particularly plant matter like that after deposition and preservation. Usually what we have to do is some chemical profiling, gas chromatography, or mass spectrometry to really understand the chemical composition of the preservation of these molecules. And here we're just, again, metasequoia. This is from the wood, the bark of the tree itself. And in this top, don't worry about this, how confusing these lines are if this means absolutely nothing to you. I'll explain it a little bit later on. But if you focus on this top figure here, this is a modern sample, a modern wood sample of metasequoia. And you can see these peaks, well preserved, obviously, it's modern. It has multiple chemical compounds, mostly lignin. Lignin and cellulose make up most of the plant matter. And in this fossil sample, you see that it's really been degraded, but you still get high peaks of some of the compounds themselves, showing that at a chemical level, this material is well preserved, even though it's 20 million years old. Not always the case, right? So this is really extreme examples. Any of these biomarkers are subject to biodegradation over time. So when you do find well-preserved sites like this, it's really remarkable because it's really our only opportunity to understand paleo-environmental history of a location. Some more modern stuff. So this was a project I had worked on a few years ago looking at the Great Wall. Well, the not so Great Wall. In Western China, they have some parts of the Great Wall are actually made of earth and rock and grasses, not the nice, beautiful Qin dynasty structure, rock structure. But anyways, this is part of the Great Wall in Western Gansu province in China. And what we were interested in is looking at, if you can see here, this is actual reeds, so water plants that were growing in the area, 2,000 years ago during the Han Dynasty. And every year, they would put a new layer down of the mud and earth, and then top it with the trees that grew, and then mud and earth, and then more trees. So we were interested to see whether we can get actual environmental changes over time based off just these plant remains, and see if there was a year-to-year difference in Western China as they were fortifying and maintaining this part of the Great Wall for barbarian raids. Preservation wasn't actually that great in this example, so it was really difficult to go horizon by horizon. But just an idea of some of the more modern biomarkers you can understand and their preservation. A lot of the times we might only have physical remains. So again, we have scanning electron microscopy. And you can see here, these are different age, Paleocene, Eocene to Miocene. Again, Metasequoia scans. And with the scanning electron microscopy, if you have this information, if you have the actual fossils, it's great to understand the state of preservation. Really well preserved. It's hard to see here, but no, not as well preserved, but enough where you can get an idea of the actual morphological features of the plant, and then compare it with modern samples. Once you do that morphologically, then you can compare it with the actual chemistry of the plant. And with that chemistry, you can get an idea of growing conditions and growing, just basically how the plant grew, and really compare it to its modern context. Again, a few more examples. These are from northern Canada, Ellesmere Island in the Arctic. And we have really well preserved, nearly all different ages, from 50 million to 15 million years old material. And again, we're really interested in why they preserve well and what this means for the growing condition, the animals that were alive at the time, the atmosphere on the entire planet, the water cycle, things like this. Again, this is SEM. You don't always have this ability to look at this under the microscope. You might need to get into a more chemical-based and only chemical-based structure, and that's what we have here. So these sediments are 15 million year old sediments, again from the Canadian Arctic, and we didn't have well-preserved plant remains or actual fossil leaves. All we had was the sediments that the plants grew in, and when they died and decomposed, what was left over were the N-alkane biomarkers. And it's kind of a messy figure if you're familiar with chromatography. This is a bit of a messy figure, but it's okay. It does show us that we have really relatively abundant peats in terms of some of the compounds that we were looking in. And this is just another way that we look at preservation. And this is what we're really interested in at Olduvai, because we don't always have the micromorphology or the microfossils to look at in terms of plant remains. But this gives us an idea of really what the climate was at the time, and what that meant for the planet or growing conditions and plant communities and vegetation. Dr. Mercader mentioned earlier some of my colleagues, Neil Gupta, who I worked with when I was studying in Rhode Island, and his former advisor, George Cody. They did some really incredible work on chitin and insect cuticles from really, really old material. So this is a modern scorpion, and they were comparing modern scorpion microscopy with 310 or so million-year-old fossilized scorpions. So in this lighter color here is what's actually remained from those 300-million-year-old scorpions. And then the epoxy, this layer down here, is just the mounting material. But anyway, you can see, I mean, obviously it's really well degraded compared to anything modern. But the fact that anything remains at all, and that there's actual organic residue left over at this age, is really remarkable. And this means a lot in terms of what we know about paleontology or molecular paleontology and what we can understand about the past. This sample here is actually even older. This might be some of the oldest organic residues that we do know of. This is 400 million years old. And what we think why it's so well preserved is actually the lipids. So upon death, lipids helped preserve the material as they're resistant to degradation and decomposition. So the lipid material from the chemistry of it is actually really well preserved. And you have the actual visible photograph, but then when you look at the chemical composition, it too is really well preserved. So what does this mean for Oldobai? So Oldobai obviously is much younger. We're interested in stuff from 2 million years to more recent. So not 400 or 300 million year old stuff. Well, there's been a few instances looking at lipid biomarkers in the archaeological record, and this is really only recent. Obviously, there's tons of phytolith data out there for bed 1. But we're interested in looking at N-alkanes and lipids and what they meant for human evolution over the last 1.8 to 2 million years. Just kind of a recap. So we are interested in the isotopic composition, which we find in the N-alkanes. But of course, there are always some issues. Once they are deposited, there always is decomposition. And the abundance you might find them preserved, but there might not just be abundant enough to do anything with them. You might be able to find the N-alkanes, but the isotope value might not be enough. But we are interested in the different types of evidence that they can provide in terms of water availability, atmospheric temperature, precipitation, humidity, different things like that. And there are a few different sites that we've really been interested over the last 2 years. And you're probably, for those in paleoanthropology or archaeology, you're probably familiar with the Frida-Leaky Gorongo site. It's adjacent to FLK, where Paranthropus boisei was found 56 years ago now. But we're looking at a few different things, both in a vertical column for temporal scale, but then also horizontally. So these ones here, we're kind of doing a broad landscape understanding of the paleo environment to see whether there are actual unique microhabitats within the gorge through time that are still uncovered in terms of their preservation, their biomarker characteristics. Now, again, don't really worry what these figures mean right now. These are some preliminary results of the chemical compositions of sediments, and they're really messy. They shouldn't look this messy at the bottom. There might have been an issue with some of our equipment itself or some of the inventory that we were using with, but that issue should solve itself. What you should take away from these, so this is FLK West here, is that there are well-preserved N-alkanes biomarkers in the sediments, and that's good. So from here, we can then go analyze the isotopes. And when we do the isotopes, we really get an idea of paleoclimate. And again here, SHK is another part of the gorge, another site we work at. And again, really messy at the baseline. That can be cleaned up with some chemical compounds and solvents. But again, we do have some nice residues left over. And like I was saying before, so I mentioned a few sites. We're really interested in the western part of the gorge. So here's the main central part of the gorge with the confluence. The PTK DS site, which I think was mentioned earlier, 1.8, and then the FLK sites here. TK, which everyone from here should know about the TK site, and then the BK and the SHK site. So again, we're interested in both preservation throughout the gorge at a horizontal scale, but also through temporal, through time, to see what the preservation was. Okay.